I, I think more attendees will come by. It's sort of plateauing out, so we'll start anyway. Right.
Sounds good.
All right. Welcome everyone to the second seminar in the Advanced Imaging series. It's my pleasure to have an old friend, Erdinç, here. Erdinç did his bachelor's from Istanbul, after which he came to Petrashvili's lab in Dresden to do his PhD, which is where we met. We played a lot of football games. He's a very good musician. After that, he continued to do, explore membrane biology with Kai Simmons, whom you might know if you're working on membranes at MPI Dresden. And he did a postdoc then with Christian Egeling at Oxford. He was also an EMBO postdoctoral fellow and a Marie Curie postdoctoral fellow. And a Newton fellow, so a lot of fellowships that he's acquired through his years studying membranes, and now he's an assistant professor at SciLife Lab at Karolinska Institute, and is probably deciding whom should be getting the next Nobel Prize there. Right? I'll take over from here, and your screen is shared, and off you go. Thanks.
Thank you. Thanks so much, Senthil. Thanks for reading from my website. It's very good to see you, and thanks for inviting. So today I'll talk about what I have done last couple of years in Oxford, as well as what we started here and what we are doing here. So as Senthil mentioned already, the focus of our research is plasma membrane. So we're always interested in what is the structure of the plasma membrane, how the plasma membrane contributes to the signaling and the cell physiology. And when you see the textbooks plasma membrane, you basically see just two parallel sheets. Sometimes they see some balls so that they would represent the lipids. But last couple of years, last 2-3 decades actually showed that plasma membrane is not an homogeneous background for proteins, but also it has this heterogeneous structure, because we have more than 1,000 types of lipids per cell in mammalian systems. We have all the proteins, and these lipids and proteins, some of them, they have different affinities for each other. Saturated lipids, unsaturated lipids, they make like different type of proteins. And all this complexity makes a heterogeneous structure where we have clusters, domains, different complexes. And this heterogeneity is not a pure functional, pure consequence, but it's a functional heterogeneity. So signaling and physiology of the cells are actually affected by this heterogeneity. And on the other hand, if you think of the membrane, we have individual components as lipids and proteins, but if you think as a collective entity, it has its collective properties, such as rigidity, viscosity of the membrane, the curvature, tension, deformability of the cell. These are very much related to the cell membrane collective properties. And this is also important because each membrane in our cell type is actually different, and this composition actually determines the collective properties. And these properties can change from healthy to disease or from cellular states. And that's why actually this biophysical properties, collective properties, can also be used to understand the state, the health of the cell. So in the lab, we're investigating this. Basically, we try to develop methodologies to visualize the plasma membrane structure and the biophysical properties of the plasma membrane. And we try to reveal how the plasma membrane structure and the biophysical properties modulate cell signaling, and eventually how these properties contribute to diseases, and if we can somehow correlate with some of the plasma membrane properties with diseases, and even try to change these phenotypes by interfering with the membrane. So, of course, this is central. The plasma membrane is the hub for signaling events in the cell, and it is the first contact of any kind of ligands that is going inside and then starting any signaling. But we still have main caveats why we cannot understand all these things that I just mentioned. First caveat is because we don't have robust probes and technologies. And if you look at the membrane, it is quite a robust structure, but if you're thinking about the lateral heterogeneity as well as biophysical properties, we don't have enough resolution, both in time and in space. Most of these clusters and domains, they form and dissociate quickly, so we need to have high time resolution. as well as high resolution in space because these entities are very small. And the third challenge is the complexity. If you think about a mammalian plasma membrane, as I mentioned, there are 1,000 types of lipids. On top of it, there's glycocalyx proteins. Underneath, there's cytoskeleton. So it's very, very challenging to understand the role of each component in the plasma membrane physiology. So to overcome these challenges in the lab, we are doing a few things. So we develop chemical biology tools to visualize better the plasma membrane and the physical properties of the plasma membrane. And we develop super resolution imaging and single molecule spectroscopy to understand the plasma membrane's properties in the nanoscale. And of course, we have to do software development to analyze this kind of data. And to overcome the complexity problem, we use synthetic biology and minimal cell systems. And to get the fingerprint of the biophysical properties that we're investigating, we do lipidomics and other omics technologies, so we can do actually comparative omics. So today I will talk about a few things. I'll start with chemical biology, briefly mentioning how we use super resolution microscopy with single molecule, and then I'll end up with the synthetic biology tools that we are developing. So the first thing is try to understand the plasma membrane biophysical properties. And this is not a very easy task because plasma membrane is such an interesting structure. It is actually a liquid structure, but it has so many components and so many parameters that are depending on each other. And that's why we cannot get a single parameter isolated from other parameters. So we need really good probes to actually get quantitative single parameters. So one of these is membrane rigidity and viscosity. These properties are very much related because if you think about the heterogeneity of the plasma membrane, we have saturated lipids and unsaturated lipids. And saturated lipids, when they form a membrane, they come together really tightly and they form this ordered, more like tightly packed membrane. And if unsaturated lipids come together, because of this unsaturation, they form a kink. And due to this kink, they cannot really pack well. That's why they form more loosely ordered membrane. So this is called ordered membrane. This is called disordered membrane. But what we need to know here, this membrane is more rigid than this membrane. And this is an important task because membrane rigidity determines how molecules diffuse, and how molecules diffuse depend how the reactions will take place in the plasma membrane. So, which means signaling output will be affected by the rigidity of the membrane. That's why it's an important task to understand what is to measure in a quantitative manner the membrane rigidity. So there are a couple of ways to do this, but one really easy method is to use smart environment sensitive probes. One of these probes is called LORDAN, for instance. And this LORDAN is a very interesting probe because it senses the polarity of the environment. If you put this probe in a very polar environment, it shifts its emission to more red spectral region. And if you put in a more non-polar environment like chloroform, it puts more into the blue region. So it doesn't have this fixed spectrum like Alexa does. It changes its emission depending on the polarity of the environment. And how are we going to use this for membrane research? So if you think about this ordered and disordered membrane again, ordered membranes are saturated lipids. They align really well. They don't leave a lot of space for water molecules. But if you think of disordered membranes, because they are very loosely packed, there's a lot of space for water molecules that would go into the interface in the membrane. And that is why these membranes, which is composed of unsaturated lipids, are more polar than the ordered lipid phases. And that's why, for instance, LORDAN has a maximum emission of 440 nanometers in the ordered domain and 490 nanometers in the disordered membranes. And this spectral shift can be used to measure the rigidity of the membrane. In a cuvette, that would be very easy, right? So just put your sample and then measure the spectrum and get the two wavelengths, this red shifted and blue shifted wavelength. And ratio of this wavelength, more or less, would give you how packed the lipid membrane is. But what we want is to understand in the cells how this is organized in the cellular membrane. So we cannot just put the cells in a cuvette and then measure. We want to see the spatial organization. That's why we need to come up with an imaging technology that we can measure single cells and still be able to get this spectrum. So that's why we applied spectral imaging with the environmental sensitive probes basically we get the emission from the sample. With the help of a prism, we disperse the signal, and with the 32 channel detectors, for instance, we can actually have each wavelength can fall into one detector, so that you can have different signal from different wavelength for each channels, so that you can reconstruct a spectrum for each pixel. And from this image, you can basically measure an empiric lipid packing parameter or rigidity parameter, which we call generalized polarization. So it changes between +1 and -1. And if it's +1, it's super ordered. -1 is very disordered, but real cells will basically change in between. As you can see here, this is some red shifted cells. This is more blue shifted cells. You can see this two cell type is basically different in each other. This is more disordered. This is more ordered. But there was one problem with LORDAN. LORDAN has been used for maybe 30, 40 years now, but we couldn't measure the plasma membrane alone. It always gets internalized. You can see the plasma membrane, but it basically labels everything inside the membrane, inside the cells as well. It goes into the membrane, it goes into ER, wherever there is a membrane, LORDAN goes into. So that's why we developed this new probe, which is called Pro12A, which has a charge group so that it doesn't internalize, so that we can visualize the plasma membrane exclusively, so that it allows us to actually understand the plasma membrane better, because now we have high contrast. And on top of this, because of the spectral region that this dye is excited and emitted, we can actually combine it with plasma proteins, fluorescent proteins like mCherry, mRuby, RFP, so that now we can understand the plasma membrane physical properties in relation to existence of subproteins and small domains where these proteins are enriched or depleted. So this probe we just published this probe, and now we're using this probe to understand the plasma membrane in a high throughput manner, because now we can actually see only the plasma membrane and we don't have to deal with the internal signals anymore. So using the smart environment probes, just with confocal mode, confocal microscopy, we can, of course, relate to many physiological conditions. So first thing, we collaborated with a group in Vienna, and we figured out that actually aging changes the plasma membrane rigidity. As you age more, your cells, especially muscles, become more and more rigid in the plasma membrane. Likewise, one of our focus in the lab is lysosomal storage disorders, where lipids are not trafficked properly to the plasma membrane, especially cholesterol. It gets stuck in the late endosomes or lysosomes. And that is why it could be an interesting target to see how the plasma membrane rigidity changes, and if this could actually contribute to the physiology of the disease. And we could actually see the disease cells are less rigid than the wild type cells. This GP unit, it's basically membrane rigidity. If higher means more packed, lower means more fluid and less ordered. Similarly for signaling, we also try to understand how, first, the signaling changes the plasma membrane. Second, how the plasma membrane changes the signaling. And here we also saw, for instance, Wnt signaling, which is a very important signaling event that is taking part in proliferation, division, even cancer, for instance. And we show that actually it disorders the membrane once the signaling starts. These are just a summary what we are doing with confocal microscopy and spectral imaging, which is with the smart probes. But this is not actually our interest, because what we are trying to understand is the nanoscale plasma membrane organization. And for this, confocal is not enough. Because if you think about it, if you put an infinitesimally small particle on a confocal microscope, in a fluorescent microscope, you get more or less half of the wavelength, which is 250 nanometers, right? So if an entity, an organization, an interaction in the plasma membrane is smaller than this, we cannot really access this. So what we did, we applied super resolution microscopy. And one of these super resolution microscopes is stimulated emission depletion, STED microscopy. So in STED microscopy, I'm not going to go into detail, but once you get the emission of your whatever sample you have, this is half of the wavelength, let's say, because it's diffraction limited. But central spot would probably be where your molecule is. and the periphery is what you want to get rid of. And that's why in the STED you have a depletion, a second laser that can actually deplete the emission that comes from your sample. And if you put it in a donut shape, the center is untouched, but the periphery is killed. And that is why you have the central spot, which is now 2 to 40 nanometers, compared to confocal diffraction limit at 250 nanometers. And what can you see with this? Of course, you can see a lot with this. This is, for instance, a particular pore complex that is commonly used as a to show the strength of the super resolution. With confocal, you can't see really the structure, but with super resolution, including STED, you can actually see very nicely the structure of this nuclear pore complexes. But for us, of course, this STED is great with many dyes. But for us, what was important is not only to see the structure, but also quantify the biophysical properties of the structure. That's why it was a must for us to actually apply STED with the smart probes instead of fixed fluorescent probes. And that's why we applied. We actually collaborated with chemists to make us a couple of dyes, and they made a lot of dyes, but only a few worked. But the ones that worked actually were very rewarding, because if you see in a confocal, this is a phase separation, two phase coexistent system where there should be nanodomains. But here in confocal, you cannot really see a nanodomain. You see some kind of greenish, bluish transition, but you cannot really be sure if there's a nanodomain or not. But here, in STED mode now, with this environmentally sensitive probe, you can clearly see here there's a blue domain, and then rest of the membrane is more ordered than this disordered domain. And of course, seeing is easy, but now with this probe in combination with the super resolution, you can not only see it, but also calculate and measure the biophysical properties like the rigidity in a quantitative manner, right? And so, of course, we realized that this is not only used to see nanodomains. For instance, endocytic vesicles that are just budding from the plasma membrane. That was also a big challenge for us to see how ordered they are. Are they the same rigidity as the plasma membrane or not? And confocal is not easy. It gives already hints that actually plasma membrane is more ordered, but we couldn't resolve single vesicles. But with STED mode now, you can see the plasma membrane nicely, single vesicles nicely, and you can now calculate the rigidity, this generalized polarization, and you can see the difference between the plasma membrane and the vesicles. You can see, probably appreciate a bit better in this picture where there's very crowded vesicles, but here you can see plasma membrane and the vesicles separately and quantified. So far, this STED technology helped us a lot to understand the lateral heterogeneity in the plasma membrane. And this is thanks to this depletion scheme, the donut depletion scheme. If you look from the profile, this is the Z, this is X, this is made by a phase plate. This is the basic trick of the STED. This is the depletion scheme. And thanks to this depletion scheme, instead of confocal, which is diffraction limited, you are seeing a small particle as close to its size. Instead of 250, now you see 220 nanometers. But in Z, the resolution is still limited to diffraction, which is around 1 micrometer. This is confocal, this is STED. There's no change at all if you use this depletion scheme. But if you use another depletion scheme, which actually gets rid of the signal on the bottom of your observation spot, instead of this Z resolution, which is 1 micrometer, now you can get around 100 nanometer Z resolution. So it is similar to TURF resolution, but this is not limited to the surface, glass surface. So you can have this 100 nanometer resolution in every plane. So that was very advantageous for us because we are very much interested in the plasma membrane dynamics, especially in the cell-cell contacts, where it contacts a surface or it contacts another cell. So here is, for instance, if you look at a cell from the profile, this is XZ scan, right? So this is the Z profile of the cell. You don't see much. There must be membranes here, and then this is the nucleus, this is on top of the nucleus, this is membrane staining. But if you look at with the 3D STED, where you increase the 3D resolution now, you can see very clearly the bottom membrane and the top membrane. So whatever you put on the surface now, you can actually understand how whatever you put on the surface can affect the bottom membrane, but you can see the changes in respect to the top membrane. So what else you can do? Of course, I told you already that we are interested in endocytosis and how the endocytic vesicles look like. Now we can really see clearly in the XZ scan the vesicles that are just budding from the plasma membrane and study them in great detail. And of course, one of the aims was to apply this with the polarity sensitive smart environment probes, and that's what we did. In confocal, you cannot really distinguish again two membranes, but now we can actually see two membranes and quantify the physical properties of these two membranes thanks to 3D STED, where we increase the resolution in the Z dimensions. So for now, I want to conclude this session. So I hope I could convince you that smart probes, environment sensitive probes, are really good tools to show the biophysical properties of the cell membrane, which changes with different physiological problems like aging, diseases, and signaling. And especially if you combine these smart probes with super resolution imaging, then you can get the nanoscale biophysical properties that you couldn't get with normal confocal diffraction limited microscopy. So in the second part, I will focus a bit on the single molecule spectroscopy tools that we are developing. So I'm sure you are very much aware that diffusion is very important for life. Diffusion itself is basically life. If you stop the diffusion of organelles or cells, the cells die. So the whole cell diffuses around, all the organelles inside the cell diffuses around, actin inside the cell diffuse, or lipids in the plasma membrane diffuse, right? This is how lipids diffuse in the plasma membrane. So everything is diffusive in the cells. And this is a good indicator of the function because diffusion determines the interaction dynamics, and interaction dynamics is basically one of the major parameters of the function of the proteins and lipids. So how we can measure the diffusion then? So in the cells, there are a couple of ways like FRAP, single particle tracking. The methods that we use is fluorescence correlation spectroscopy, mostly because it's historical, like Senthil and I, we did our PhD with Petroshvili, who was an expert of fluorescence correlation spectroscopy. And here, what we do, we just park our spot and we let the molecules, fluorescent molecules, come in and out. And this molecular diffusion creates fluctuation. And from this fluctuation, we basically get a correlation curve by applying a function. And from this correlation curve, from this fluctuation of the molecules going in and out of the focal volume, you can get the diffusion time of the molecules that is actually going through the focal volume, which is the diffusion time that the molecule takes to go through the focal volume. But of course, this is definitely a very useful tool because now we can apply this technology in cell membrane, for instance, to see the diffusion of lipids or proteins in the plasma membrane. So for instance, we check how different lipids diffuse in the plasma membrane of cells, zebrafish, like organism level, you can also do it. You can check how proteins move in the nucleus. We are not only limited to plasma membrane, but you can do this diffusion measurements, any compartment in the cell basically. And you can even go a bit smaller in the structure. We also did FCS measurements in bacteria to see the protein complexes in the bacteria and how they actually pre-assemble in the cytoplasmic space before they go to the membrane. But again, the important thing is we are after the nanoscale, right? So the confocal based FCS is cool, but what about the nanoscale properties that we cannot access? The plasma membrane heterogeneity, for instance, that we are looking is in the small scale. If you look with the big confocal volume, which is 250 nanometers, you average out all this complexity and you get one single value. Instead, if you make smaller observation spot, like with STED, for instance, now you can actually get the domains and the diffusion of this, let's say, the heterogeneity in the plasma membrane will be more represented when you have a smaller scale observation spot. So what is gonna give us? So first of all, if you have a small spot, you can do diffusion measurements on structures that you couldn't do before. For instance, you can do diffusion measurements on viruses, on organelles like peroxisomes that you couldn't do before, because for FCS, you need molecules to go in and out of the focal volume. If the focal volume is bigger than the structure, you cannot do FCS. The second thing that we are more interested is the diffusion law that is shown here, that I want to show a bit more in detail. So if you change the observation spot size from confocal, which is 250 nanometer, and you make smaller and smaller in terms of the spot size, if the diffusion coefficient somehow doesn't change, this means it's a free diffusion, the Brownian motion that we always think when we think about the diffusion. But there are different diffusion modes. It's not only free diffusion in the cells. Cells actually could have different diffusion modes, and we can access this by changing the observation spot from confocal all the way to super result. And if the diffusion coefficient goes down, we call it a trap diffusion. There's a lot of simulations and a lot of models behind it, that if molecules diffuse and transiently trapped in one spot, the diffusion coefficient should go down. If they are transiently interacting with a domain that moves, then the diffusion coefficient should go down, and it should level out at some point and stays constant. And if the diffusion coefficient is going up as you change the observation spot smaller and smaller, then this is the Hopf diffusion, which is very much related to actin that we are going to discuss a bit later. So all this, first when this came out, this molecular diffusion law, it is called, or let's say diffusion mode, it is beyond if molecules move fast and slow. It actually tells you how the molecules move, not only fast and slow, but its real nature, also its physical principles, why molecules move fast or slow. And of course, we set out to test how different molecules in the plasma membrane move. For instance, phospholipids, cholesterol, sphingolipids, gangliosides, proteins. We tested all of them, and now I'm going to show you a few of them that actually shows the real heterogeneity of the plasma membrane. So if we tested phospholipids, we either saw a free diffusion, the diffusion coefficient doesn't change as you go from confocal all the way to 50 nanometer stat resolution, for instance. Or you see Hopf diffusion, where the diffusion coefficient goes up as you go smaller with the diameter. This is phospholipids, but if you go to sphingolipids, very specifically sphingolipids, you just see it goes down, the diffusion coefficient goes down as you go smaller and smaller with the diameter of the observation spot. And gangliosides, for instance, GM1, which is very important for neurodegenerative diseases, for instance, it shows kind of domain diffusion. It goes down a bit and then it stays constant after a while. And we were, of course, wondering what is behind all of these diffusion modes. And one of the candidates is, of course, actin cytoskeleton. To study actin, there are a couple of ways. You can use actin drugs, for instance, but we wanted complete absence of actin. That is why we treated cells with some vesiculants, and the cells bleb out and form these vesicles, which we call giant plasma membrane vesicles. So these GPMVs, giant plasma membrane vesicles, they bud from the cells, so they're almost, the composition is almost the same as the plasma membrane, but they don't have cortical actin cytoskeleton. If you label actin, it forms this rim underneath the cell, but in GPMVs, they're only monomeric actin and there's no organized cortical cytoskeleton. So this was a good system for us to start testing how this diffusion modes change, and I'm gonna just show a summary where we tested everything that we tested in cells, also in GPMVs. So all this Hopf diffusion that we have seen, they're all gone. So Hopf diffusion is definitely a parameter of actin cytoskeleton, which was already predicted by several groups, including Aki Kusumi or Jitu Meyer. So sphingolipids, the trap diffusion is also gone. So this trapping, whatever sphingolipids are trapping with, they're also gone when actin is gone. But the gangliosides, that's not true. So ganglioside domains, they're still staying intact, even if there is no actin cytoskeleton. So with this, I'd like to conclude this part. So the cellular diffusion, this is quite an important parameter that we actually keep forgetting. So it is an indication of function. If the diffusion of a molecule changes, its function might also change. And STED combined with FCS is a very good tool to access the nanoscale diffusion. And the GPMVs are very useful tools if you don't want any actin present with almost the complexity of the plasma membrane. And the actin cytoskeleton influences diffusion of most, but not all membrane components, such as the gangliocytes. So with this, I'd like to briefly mention synthetic biology tools that before I finish. And here we are basically trying to understand the role of each component on cellular processes that happen in the plasma membrane. But if you think of the plasma membrane, we have all types of lipids, cytoskeleton, glycocalyx, proteins. It is very difficult, especially for lipids and sugars, it's very difficult to get rid of one component at a time. For proteins, it is relatively easier. You knock out the protein and you see the function of the protein. But for lipids and sugars, it is very complicated because one single enzyme does many different jobs. So if you knock out one enzyme, you're not influencing one lipid or one sugar, you're influencing a lot of different lipids and sugars. That's why we go a different way. We use the bottom-up approach. So in the bottom-up approach, we try to reconstitute from scratch. We start from lipid systems, and then we add proteins on top, we add the sugars on top, we try to reconstitute actin and extracellular matrix so that we can understand the role of each component, how they influence the cellular processes at the membrane. And we, of course, use all of these to understand different metabolism-related problems, immune signaling, host pathogen interactions, and various other questions. So here I'll just show you one simple system where we use lipid-only system, try to understand the metabolism of lipoproteins. So lipoproteins are quite central to our health because they're basically carrying lipids around the body. So LDL basically gets cholesterol and other lipids to the body cells, and HDL collects excess cholesterol from the body back to liver so that it's recycled. So the balance between HDL and LDL is quite important. So if you have more LDL, that's called bad cholesterol. So you have hypercholesterol. And this uptake of LDL and HDL is basically via receptors. So LDL has its own receptor, which is called LDL receptor. HDL has its own receptor, which is called SRB1. So this basically, those receptors, they have interplay. HDL and LDL levels should be regulated very tightly. But there is one thing, there is this very frequent disease called familial hypercholesterolemia. It is quite frequent, as 1 in 200. And here, the LDL receptor is mutated. And that is why there is aberrant LDL trafficking. That's why you have high LDL amount in the blood, and it leads early cardiovascular diseases. But although these patients don't have LDL receptor, they still somehow get cholesterol to their membrane. So there must be another way how they get the LDL to their membrane, the lipids to their membrane, to their body cells. And that's why we ask if LDL can interact with the target cells without the receptors. So for this, we have a lipid-only system. What we do, we just deposit some lipids on an electrode, we apply electric field, and we get these vesicles, which we call giant unilateral vesicles. You can get these vesicles basically labeled with fluorescence. You can study them with a very tight compositional control. So you can determine the lipid composition perfectly. So here, what we do, we get the lipoproteins from the patients or healthy donors. We characterize them by AFM or other single molecule technologies. After we see their size and shape is properly representing the lipoproteins, we see their interactions. For instance, applying cryo-EM, we see these lipoproteins, different lipoproteins, and we see they actually interact with our liposomes, our synthetic cells. And if you label these lipoproteins with fluorescent cholesterol and fluorescent protein, because they also have proteins on the surface, you can actually see they can transfer their cargo, which is cholesterol, and the protein onto our synthetic cells, which is this big, large, giant unilamellar vesicles. So it means even if they don't have the receptor on the surface of these lipid vesicles, they can deliver their cargo without the receptor itself. And this is quite affected by the lipid composition itself. If the lipid composition gets more and more rigid, the delivery gets less and less efficient. So it's basically, it makes a lot of sense because it's, if there's a rigid membrane with high cholesterol, you cannot deliver cholesterol anymore. So there's no delivery anymore. And of course, this lipid system is quite cool to understand how lipid complexity changes cellular processes. But at some level, you need proteins as well. You cannot do everything without the proteins. So here we also have a way how to click the proteins onto surface, especially if you're dealing with the extracellular part of the protein. You just have your vesicle. You can put some of this nickel NTA lipids with nickel NTA presented on top of the lipid. And if you use a HISTAC protein, you basically cover your vesicle with a HISTAC protein, and your protein now is presented on the surface of your vesicle. So now you have a synthetic cell with a perfect composition, tightly controlled composition of the lipid, as well as the proteins. And if this protein is, for instance, recognized by another cell, your vesicle will be kind of a cell that would interact with another cell. For instance, we study immune signaling. If you put a T cell on top of the synthetic cell where you have an adhesion or a signaling protein, T cell recognizes the vesicle and then forms this adhesion, which we call synapse. And this is quite a dynamic synapse. You see this in immune cell. This is a synthetic cell covered with one single protein that the immune cell recognizes as a threat. So that's why immune cells are trying to act against this synthetic cell. And what we try to see with this system, just to show you what we are working on. So we work on two signaling events, T cell receptor signaling, which is the major fight against the tumors in the body, and Fc epsilon receptor signaling, which is the main allergy pathway. So these two pathways, they're quite similar in structure. They have receptors that needs to be phosphorylated so that the signaling becomes active. And here are the players. Basically, we have antigens that would be recognized by the cell, and we have a phosphatase, which basically blocks the signaling. It is the red light of the signaling. But somehow, when two cells find each other, this phosphatase should be neutralized so that signaling starts. And we try to understand how it's neutralized by checking the protein distribution in the contact zone. And for this, we reconstitute our systems. We put proteins of interest on the surface of the vesicles. And what you see here, the purple, the magenta, is basically the adhesion plus the activating signal. Wherever you see the magenta signal, which is the activating signal, you basically see the exclusion of the green signal. So green protein is not there anymore because it's excluded from the contact. So here you can see a bit more clear. You see the receptor, which is the activatory signal, which is another protein, which is also in the contact zone. But what you see is complete exclusion of this red light, which is keeping the cell in the resting state, is gone from the contact, which means now cells can actually activate through this contact zone. And we figured out it's actually size. This was postulated before, that size of this molecule could be important. So when we made a smaller size of this protein is not excluded anymore. And now the signaling is completely gone. So we tested with different methodologies, including biochemistry. So these cells don't signal anymore if you shorten this extracellular portion of this single protein. So I don't want to go into detail. I hope in this part, I just wanted to tell you that we now can reconstitute the cellular processes that happen in the plasma membrane in a 3D environment, in an isolated system. And more important take-home message from this immune signaling is this physical parameters that we usually ignore, like the size of the protein, the charge of the protein, can actually be very important for the signaling activity of the protein. So with this, I'd like to thank all of the people that contributed this work one way or the other. I'm not going to go name by name, all the funding bodies. Thank you very much for listening to me, and thanks Senthil for inviting me here again.
Fantastic talk, Ardinch. Great to see how gone so far from just membranes, synthetic membranes, to all these biology-related questions. Also, thank you very much for staying awake at 1 a.m. and accommodating our Australian time zone. I'm gonna ask the questions that are anonymous, and then I'll also allow the questions that have names on them to talk so that they can ask themselves. So there are two anonymous questions which are regarding the basics of STED. Can you explain a bit more on how do you achieve Z-STED? How is the second depletion beam being arranged? How do you get the XZ scan to get the XZ image? Is it achieved by some specific type of camera? Do with the scanning thing.
Yeah, so the first part, so this depletion schemes can be arranged in a couple of ways. The first, more primitive way was a phase mask, right? So the phase mask basically forms a zero, and you can actually arrange the phase mask so that it forms the zero in the Z dimension instead of XY. Actually, the Z-STED was one of the first STED that came. It's not really novel that we just applied it to membrane to understand the membrane better. The second way to make the depletion that we used in these papers is basically with the spatial light modulators. It is, imagine that you actually bend the light so that you can form somehow a zero intensity at one place and a depletion scheme in the other places. So mainly the spatial light modulators is what is used mostly in the contemporary microscopies. The second, if we need special detectors, no, it's XZ scan is now built-in property in most of the commercial systems as well. Instead of XY, you just select XZ so that it makes one scan and then moves a bit and another scan, moves a bit, another scan. So this is basically, if you have any kind of scanner-dependent microscopy, I am sure you can do XZ scan like XY scan.
Cool. Next question is from Mike Ryan. Mike, do you want to ask the question?
Sure, Senthil. Thanks very much. That was a really lovely talk. I'm really interested in this idea that the actin cytoskeleton is modulating the phospholipid mobility. And I'm just wondering about how you perceive that to happen. Is it really about the actin cytoskeleton or making some sort of mechanical stress on the plasma membrane or curvature, and that imparts that decreased mobility? Or is it something else that we're looking at here?
Thanks, Mike. Thanks. This is actually a good question because so the simple answer, although there is a lot of work on this, we still don't know exactly. But I would go with the generic answer, which is there are a couple of hypotheses. The first one is being the picket fence model, how actin organizes the plasma membrane proteins kind of picket-like, so that you can have your fences and compartments just driven by actin interactions of the transmembrane proteins. So transmembrane proteins interacting with actin forms this mesh so that your molecules that are trapped inside the zone is moving freely. And when they want to pass from one compartment to another, they have to slow down, pass, and then move freely again. So this would explain the Hopf diffusion perfectly. So this is the main idea of Hopf diffusion. So second part is mostly the trapping diffusion. So actin pinning points are quite important for us because actin somehow definitely changes the collective behavior of the plasma membrane by pinning a lot of pinning points. Some parts, they are definitely immobile. So these parts could actually explain how this transient immobilization happens in this actin pinning point. But this is purely hypothetical that I'm speaking for this part. So the parts that actin don't change, it means just lipid-lipid interactions, lipid-protein interactions, just spontaneously happening and forming some kind of domains. But there's two main mechanisms. One, pinning points making immobile parts, and the second is the picket fence model where actin forms fences would probably be mechanisms that would explain.
Okay, thanks. Can I ask the other question, Senthil?
Sure.
Which is really about nomenclature, and you're talking about nanodomains and other areas. You don't call them rafts anymore. And I know this has been a hot topic in the past, but is this something that you avoid using that word because it's just so controversial in the field, or is there something else?
Yeah, you got me there, Mike. I think I mean, raft idea, I would say, I think it's still valid that we have this heterogeneities, but I think raft idea was, it was given a different meaning of one subtype of the heterogeneity. And when we talk about different heterogeneities, I think raft is not inclusive enough to talk about what we are seeing here. So all these clusters, for instance, this PIP2 clusters. We also see this domain diffusion for PIP2, but clearly PIP2 is not in rafts, right? So when it comes to this nanoscale properties, I think rafts especially is a bit exclusive term, not because rafts were like this, but we gave this meaning to rafts historically. So that's why I'm not actually using rafts anymore.
Thanks for that.
Very diplomatic answer, Ardinch. I was looking for Sarah Keller, who was attending, and I wanted to pull her into the discussion, but looks like she escaped.
Oh, thanks God.
Next question is from Alex Coombs. Alex, do you want to ask?
Yeah, thanks. Great talk. Look, I had some technical questions about whether the rigidity measures work in fixed tissues or live tissues. When you actually got cells abutting each other, and it's really difficult to determine which membrane belongs to which cell. But I guess the broader question that I'm interested in is whether you think that membrane rigidity plays a role in tissue morphogenesis, so during dynamic changes during development.
Mm-hmm. Yeah. So I think this is definitely happening. So we collaborate with a couple of developmental biologists on this. One being Marianne Bronner from Caltech, and with her postdoc, we looked at how migratory cells are different from non-migratory cells, and clearly it's different, and it makes sense. Rigidity is telling how much the cell can move. So we can definitely measure those things. In the embryo, yes. In living systems, yes. Ex vivo, particularly, explants, that is very easy. But in fixed cells, that is not very easy, because these measurements are based on the hydration in the cell membrane, which is determined by cholesterol, saturated lipids. As soon as you fix the cells, you don't have this native property anymore. So I think in fixed cells, that is probably not a good idea.
Okay, yeah, that's really interesting. I'm looking forward to seeing that work. I had another question, if I might, Senthil, and this was about the synthetic lipid sphere system. And so you'd introduced a protein to kind of look at how that membrane localized protein clusters, in activation. Can you also introduce other components? So you might be able to look at phosphorylation events, inside the spheres. So can you introduce other proteins to kind of use that as an assay?
Yeah. No, that's good. So yes and no. What we are working on at the moment is the extracellular parts of the protein, which is quite important for us because that's what we're interested in. But if you want to reconstitute the internal part, there are methods to reconstitute the whole protein, but it's not as easy as what we are doing at the moment. So you have to have very fancy, complicated technologies. But if you want to reconstitute only the internal part, that is also possible by easy methods. And this is done by a couple of people. So the full protein reconstitution, of course, is done, but I think it is becoming less and less done because it's very difficult and it's not as rewarding as the effort that you're putting. But a lot of people are doing this intracellular signaling. Ron Vale, for instance, published a few years ago where they just reconstituted the internal parts to see the phosphorylation events, and they showed the proteins they were constituted form kind of liquid domains, and they phase separate because of the charge, and phosphorylation being the main driver of this phase separation. So it is possible, definitely.
Okay, thank you.
Cool. Ardinch, there's another question. In bottoms-up approach, as you started from lipid bilayer, is that equivalent to model biomembranes? I guess the confusion about what synthetic bilayer is, or GUVs or supported lipid bilayers. If you can expand on that.
So can you ask again? I didn't get the first part.
So I mean the question is sort of phrased weirdly, but if I understand the question, it's asking what does bottom up approaches mean? What is the bilayer there?
Yeah.
Is it from a cell or completely from a point of view?
Yeah. So bottom up means we start from scratch, nothing, and then we start with lipids, and then we add proteins, and then we add whatever we need, one component at a time. In contrast, the opposite of it is top-down, which you start with a cell, and then you delete one component at a time. So top-down, bottom-up approach, they're quite complementary, but for us, for lipids and sugars, top-down is super challenging because the enzyme, the promiscuity for lipids and glycocalyx especially. So that's why we start from scratch and build one by one.
Cool. There's another ambiguous question, and I'll try to make sense of that. In the diffusion mode, what does the diameter mean? Diameter of what? So I guess this is pertaining to the spot of the excitation from spot variation. So if you want to expand on that.
Yeah, maybe I wasn't clear enough. Yeah, this is the diameter. It's not the diameter of what. It's not the diameter of the object we're looking at. It's the diameter of the observation spot. So diameter of the observation spot, which we can call also full width half max, is half of the wavelength in confocal, and we shrink it by STED light gradually. So instead, we don't have super resolved, just two steps, confocal and super resolved. We can actually change the STED laser and gradually tune the observation spot we are looking at. So that is the diameter that I'm talking about.
Thank you for the explanation. I just had one question. In the immunological synapse, where you could decrease the size of the protein and make it escape into all the plasma membrane, is it following along the same concepts that Dan Fletcher published about molecular sieving sort of thing? Is that?
No, that is, exactly. I think we basically submitted our papers at the same time. They were looking at macrophages, we were looking at mast cells, and it's showing exactly the same thing. One with allergy signaling, histamine release, and Dan Fletcher was looking at the phagocytosis with macrophages. But it's exactly the same concept.
So I understand the macrophages or immunosynapses are sort of exaggerated extreme geometries where this can be observed. Do you think, or can you comment on other general cell biology relevant systems where this could be happening?
Yeah, I think it probably happens in every cell-cell contact, like cell junctions, for instance. We don't know cell junctions if they're homogeneous. How would big proteins somehow localize to those, right? So I think now people are starting to go into this more cell-cell junction. And I think you can imagine neural synapse, for instance, is another point that now people also started exploring with the analogy of immune synapse, trying to look at the size of the molecules, for instance, how they are organized on the neural synapses. So I think you can think of any kind of cell-cell interaction, and size is becoming relevant.
Cool. There's another question from Oleg Chernyavsky. Oleg, if you want to ask the question.
Yes, I just was wondering about the instrumentation you used. Did you use commercially available off-the-shelf instrument, or you built custom modified spectral STED? Thank you.
So that was half and half. So we got a barrier resolved system, because by then a barrier couldn't sell a STED system because of the patent was with Leica. But then we built a STED around it. But after it was free, after a few years, Leica couldn't hold it anymore. I mean, it was free and everyone could produce. Then we got a lot of STED parts from the barrier system. So it was a hybrid system. But now, whatever we could do in our hybrid system, you can do with any STED microscope that you can buy, including Leica, a barrier, PicoQuant. Everyone actually is doing exactly what we are doing now.
Cool. All right. I think that's all the questions we've had now. If there are no further questions, then again, Erdinç, thank you very much, especially for your 1 AM talk. And I'll send you another link to follow up and chat about other things.
Yeah,
right.
it was a true pleasure for me, Sento.
For attending as well. Erdinç, go on. You want to say something?
No, I just said true pleasure for me. You know, if you said 3 PM, I would come. 3 AM, I would come. So
that's dangerous. All right. I'll send you another link. See you later.
See you, Sento.
Bye.